What's up, Sci It's your boy Nickel here, and we're going to look at steps of mitosis. So, if you look at the framework for these pages, we're going to complete 2 pages, or at least it took me 2 pages to draw all these different steps. So, you can see we're going to draw 4 different steps of mitosis, and we'll finish the rest of the steps in the next video here. But to get started, you're going to want to go to the extras bin and pick up a little square here of the process of mitosis. So we'll use this pie chart to understand how a cell lives its life and then goes through that process of actually dividing to make another cell, because that doesn't actually happen all of the time. It's not constantly going through mitosis. A lot of the times a cell just chills in its actual state and does its job, its intended purpose to allow us to survive and thrive. So here we go. Let's get started. The steps of mitosis. Here's part 1. So as we look at this pie chart, you can see that the process of mitosis is really just a small sliver here. And mitosis, the division process, happens in this little set of mitosis. So as we're talking about each step, just realize it takes a very short period of time. It's several different steps, and after mitosis the cell goes through this phase that we call cytokinesis, which is kind of part of mitosis, which is why we say here it's in the mitotic phase. And you can see that we have one cell. It lives its life in this phase called interphase. It then goes to the end of its life, it goes through mitosis and it creates a new cell that's identical to the original cell. The original cell doesn't necessarily have to die, but it at least produces an exact copy of itself. So, as I mentioned here, after a cell is created, it goes through this phase called interphase. If you think about the interstate, you drive on the interstate to get between different states or within a state. So if we think about interphase, this is like between phases. We're not in the actual phases of mitosis yet. We're kind of between the phases of mitosis, and this is where a cell spends 80 to 90% of its quote-unquote life. Each cell might not be alive in of itself, a single-celled organism, but even a cell in our body, if we say that it's life, it means that, like, how long is it functioning, essentially. So 80 to 90% of the cell's functioning life is in this phase called interphase. And at the end of interphase, there's this phase called G2. So let's just find that real quick. It's this huge piece of the pie right here, which means that G2 is a big part of the cell's life. It's almost a third of the cell's life. But in the late stages of the G2 phase, right before the cell begins division, in this late G2 stage, we actually have something really cool happening, and that the cell knows that it's going to be dividing, and so the organelles and the DNA are duplicated. Which means that there's a copy of themselves. So we have a double set of organelles, a double set of DNA, so that when the cell finally finishes this process of copying itself, each cell has the exact same stuff that the first cell had. So in order to draw the cells today, you're going to want to have multiple colors. So you'll see me going between colors here as we draw the different cells, but we need to draw the cell here in interphase. So if you need some colored pencils, go grab some, pause the video, and I'll see you when you get those pencils. Okay, so now we're going to start drawing the cell. So I'm going to draw myself a circular shape here. And in this circular shape, we're going to have these little things are going to reproduce. So I'm going to just draw some rectangles here. These are one of the organelles that you learn about sometimes as you research cells. These are called the centrioles. Each of these yellow things is a centriole. But they work in pairs. And in pairs, they send out this little network of tubes that kind of go throughout the cell. And these are only activated during mitosis. The centrioles are. And when we look at a pair of centrioles, because they actually work as a partnership, a pair of centrioles is called a centrosome. And the centrosome organelle works during mitosis. So a centrosome is basically 2 centrioles. Okay, so we're going to watch them as they do this little dance throughout mitosis. We'll keep an eye on them. And then we're also going to keep an eye inside of the nucleus. So if I draw a circle here, that kind of represents the nuclear membrane, which means that like the nucleus is a satchel. So it's made with a fabric, and that fabric is a membrane. But since it's not the outer membrane, we call it the nuclear membrane because it creates the nucleus. So inside of the nuclear membrane, we're going to find some DNA. And the DNA here, let's do purple for one of them, is just kind of floating around in here. But there's going to be 2 copies of it here that get made because the second copy was made here in the G2 phase. So we'll have a purple one. And I'm also going to get a green one here too. So this one also has a copy of itself. And it's just kind of floating around. But an important part of this G2 phase is that during this phase, the DNA is duplicated. There's not usually 2 copies of the DNA in a cell. There's usually only 1. But in this step, DNA is duplicated. All right. So we have our centrosomes have been duplicated, and we have 2 sets of centrosomes with 2 centrioles. We have our nuclear membrane that's still there and holding our DNA in the nucleus. And then the cell is ready to then jump into the process of dividing. And so it starts in the first phase. Let's call that step 1. And step 1 is called prophase. So, prophase. And in prophase, let's draw ourselves the cell so we can show what's happening inside of here. We have our cell. We have the spindle fibers. We have the centrioles starting to actually move away from each other across the cell because they want to get to the 2 ends of the cell, which we would call the poles of the cell, like the North Pole and South Pole. And they're sending out these tubes, and they kind of actually send the tubes to each other because these tubes can push against each other, they can pull. It's basically like Spider-Man's web in here. Alright, we still have our DNA that is secure inside of the nuclear membrane, and the DNA is actually starting to kind of go from its loose, loopy shape here, and what we would call that would be the chromatin, and it starts to condense, and it starts to pack itself more and more into that chromosome that it's going for. So if we draw this, it's going to be a little bit more condensed, not fully condensed yet, but I still have a double copy, and they're both condensing of purple and of green here. So I've got my DNA condensing. Now let's label what's happening in this step. So these little lines here, as we were saying, are called spindle fibers. So we say the spindle fibers start to form. And we'll say that the DNA, which is in the chromatin state, so we'll say DNA chromatin starts to condense. Now this is basically just an elaborate dance, and so the very beginning, when the beat drops at the beginning of the song, boom, the organelles and DNA duplicate. Then the first step of the dance is prophase. The spindle fibers start to form as the centrioles move apart from each other, and the DNA starts to condense down, and then we jump into our next phase, which is prometaphase. Prometaphase is the next step, step number 2, and so we need to start by drawing our cell again. And those centrioles are actually now on opposite poles. So I have one set up here and one set down here, and they're sending those fibers all throughout the cell. And they're actually in search of the DNA. They're looking to grab a hold of the DNA. So let's see what's happening with the DNA at this point. The membrane that was holding the DNA actually starts to dissolve. So I'm going to draw it as a dashed line because it's kind of there, but it's dissolving. It's starting to fall apart. And then inside of the nucleus, the DNA is now fully condensed. And so I'm going to draw it fully condensed here. And as it condenses, the two things that are copied to each other, the two pieces copied to each other, actually find each other and they attach to one another, and they make this thing that looks kind of like an X. And we call this now a chromosome. This has now formed into the chromosome because it's fully condensed and it has the two parts to it. But remember, the left side is identical to the right side of each of those chromosomes. Alright, so let's label what we just were talking about. We have the spindle fibers here. Well, let's come back to the spindle fibers. We're going to add a little bit more to that. But we do have the chromosomes fully condensed. And we also have the nuclear membrane, which is now dissolving. But something we need to add here is that these spindle fibers actually find the chromosomes. And each side comes in and attaches to the middle of these chromosomes here. So we need to make sure that we include that, because this really sets up the process of the tug-of-war match that comes here in our next step. Before we move to the next step though, let's just label that this is happening here, and that the spindle fibers attach to the chromosomes. And then we can move on to metaphase. Step 3. Once again, let's draw our cell. So we'll draw ourselves a little circle here, and this will be the last phase in part 1. But we have the centrosomes here on each end of the cell once again, and they have sent their spindle fibers shooting out all over the cell. Some of them actually find each other here on the outside, and they start to push on each other because these two centrosomes want to get further and further apart from each other so that they can then be in their own cell. It's basically like two siblings that are fighting, like, no, you go to your room, no, you go to your room. And they want to get away from each other, but at this point neither are compromising because they don't have what they want. Each of them wants a copy of the DNA, but right now they're going to have a tug-of-war match. And so something that I'm going to start to do here to simplify these chromosomes, the chromosomes that are condensed and look something like this, instead of drawing those with the squiggly lines, the way that we typically then draw those is just like a bubbly looking X here, just to simplify the shape. So as we draw them down here, we're going to simplify the shape, and we're going to show that these chromosomes, actually in metaphase, they're going to align in the middle of the cell. And so this tug-of-war match pulls the chromosomes right here into the center. So we have our purple one and we have our green chromosome. And they're both in the middle, but they're being tugged on by these spindle fibers. Okay, so we just need to label that that is happening here, and so we'll say that chromosomes are pulled and aligned into the middle of the cell. And each side is holding on to the chromosomes because each side wants a copy. And so they're going to pull and pull and pull until we'll continue that in part 2. Dun, 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 dun.